everyone, welcome back to class. We've been talking about transcription. We compared it in prokaryotes and eukaryotes. And then in eukaryotic cells, we discussed how there's post-transcriptional modifications and how those modifications create functional tRNAs and functional rRNAs. And now let's talk about how we modify our mRNAs to have a functional transcript. One thing about transcription is that transcription is occurring in the nucleus where the DNA is. But to be able to be translated into a protein, it needs to move to the cytoplasm where the ribosomes are. And before that transport out of the nucleus happens, our messenger RNAs have a substantial amount of processing that's going to happen to them. Your pre-mRNA, your primary transcript, is going to be really long. It's going to be anywhere between 2,000 and 20,000 nucleotides because it's got everything that your DNA had in that gene. And this is sometimes called heterogeneous nuclear RNA, or hnRNA, this long piece of RNA. This is non-ribosomal RNA, non-transfer RNA. It is a pre-mRNA that is still in the nucleus, has all of this extra things in it, not yet ready to be functional. hnRNA is a mixture of mRNA molecules and their precursors, the pre-mRNA. The pre-mRNAs are going to be processed by doing things like removing some of these internal sequences. So you can see these gray sequences, these introns have been removed. We've added a cap to both the 5' tail and our 5' cap and a 3' tail. The C-terminal domain of part of the RNA polymerase II is going to be a platform for protein complexes that are involved in this RNA processing. So eukaryotic mRNAs will have a modified nucleotide that becomes their 5' cap. And their 3' ends will have a poly A tail. Poly A refers to it's just a lot of adenines added in a row. That modified nucleotide at the 5' cap is a guanosine that has been methylated. This methyl group right here. And it is going to be bound to the RNA molecule by a 5' to 5' linkage. So this 5' end of your RNA and the 5' end of this methylguanosine bind together. It's not the traditional 5' to 3' bond. The 5' cap is added pretty soon after transcription is initiated, and that will help contribute to the stability of mRNA. It will help protect that RNA from nucleases that would break it down. It plays a role in positioning the RNA on the ribosome for translation to be able to start. Then you can see we're also adding this poly A tail at the 3' end. This can be anywhere from 50 to 250 adenines added on there. And it is added on there with this enzyme, poly A polymerase. This 3' tail will help protect the mRNA from nuclease attack. So as a nuclease is around, if it starts breaking off stuff, it's just breaking off this A tail. It's going to take a while before it actually got to the actual RNA part of it. The length of the tail helps it just be stable. The tail is also required for it to export from the cytoplasm so that the transcript can move into the cytoplasm. And it might also be involved in helping ribosomes recognize the mRNAs and bind to the mRNAs. There's also little pieces of that transcript within the RNA that are going to be removed. These are called introns. Sequences of DNA or RNA that are in the primary transcript that are not going to be in the functional RNA. With introns, I think that they stay in the nucleus or they are in the way. They are going to be taken out of that RNA before the RNA leaves the nucleus. And you can do something called R-looping to see where the genes have introns. So here's your RNA, and if you base pair it along with the DNA, you see these loops occur. Those loops represent the different strands of DNA that created RNA that was cut out. So that tells you where that RNA intron was. You can see this gene has multiple loops on it, so you can have multiple introns. The sequences that are going to be in the final RNA are called exons. They're expressed. They exit the nucleus. So the introns are removed and the exons are put together into your final RNA. Introns are present in most of the protein coding genes of multicellular eukaryotes, although their size and the number of them varies considerably. The way that we cut out the introns and put the exons together is called RNA splicing. So here we're going to cut out these introns and put the exons together in this final copy. About 15% of inherited human diseases will involve splicing errors. And if you are incorrectly splicing, you're going to end up with an incorrect mRNA, which means you'll end up with incorrect protein products. And at that intron-exon boundary, there are sequences that are going to be what determines those 5' and 3' splice sites. If you analyze the base sequences of different introns, you can see that the 5' end of an intron usually starts with a GU, and the 3' end usually ends with an AG. And the sequences immediately adjacent to those ends of an intron are going to be similar. One of those is called a branch point. A branch point has an A residue in it. Spliceosomes are the molecules that help do splicing to take out the introns. And there's 5 types of RNA and more than 200 proteins involved in a spliceosome. Here, this is an actual micrograph of a spliceosome, cutting out the introns and putting the exons back together. The spliceosomes will assemble on the transcripts from starting with a group of smaller RNA protein complexes called snRNPs, or small nuclear ribonuclear protein complexes. And each of them have 1 or 2 snRNAs, or small nuclear RNAs. So those small nuclear RNAs start to assemble into this spliceosome to allow splicing to occur. Spliceosomes will assemble by binding to the snRNPs to the pre-mRNA. In the first step, the snRNP called U1 binds, and U1 is going to be able to base pair with the 5' splice site. And then a second snRNP called U2 binds to the branch point sequence. Then a group of snRNPs, so U4, U6, and U5, will bring together these ends of the intron together, and this is forming a mature spliceosome. The pre-mRNA is going to be cleaved to the 5' splice site and form this lariat structure. When the lariat forms, the 3' splice site is cleaved, and then these two ends are going to be joined together with an exon junction complex forming at the boundary of these 2 exons. The EJC is going to be required for that mRNA to be able to efficiently be exported, and also can influence things like degradation of mRNA. And that lariat of the intron is going to be removed and degraded. Some genes have self-splicing RNA introns. They're able to, the RNA itself, catalyze that splicing process. When you have an RNA that's able to catalyze its own reaction, it's called a ribozyme. Ribozyme. There you go. There are some important impacts of having splicing happen in a cell, and one of those is alternative splicing. So if I have this gene, I can have different areas become an intron or an exon and end up with alternative splicing that gives me different proteins. So this could give me here or these or different types of mRNAs. I can have certain splice sites be activated, certain splice sites be skipped. using things like splicing enhancers and silencer sequences. And by having alternate splicing, I can get lots of different versions of a protein from one gene. So in our bodies, in a typical human, you have much more proteins that you create than the number of genes that you have, because in different situations, different genes will be spliced differently to create different proteins. It's kind of a cool way of conserving energy from not making too much DNA, but still getting the protein products that we need. Introns also have a few different jobs besides just alternative splicing. They also allow new protein coding genes to be formed through recombination. Recombining different introns can make new combinations of exons. That's called exon shuffling, kind of moving your exons around. It can also make duplicate copies of exons within a gene. And by having duplicate copies, you have one kind of as a backup doing what needs to be done. But that duplicate copy could mutate into a new sequence and be able to create new functions within a cell. Then on the C-terminal domain of RNA polymerase II, this helps with a lot of RNA processing. A lot of these processing events aren't waiting until after transcription is happening. They are done at the same time, so they occur co-transcriptionally. So if you have your RNA polymerase here, you can see the capping enzyme here during transcription. You can see the splicing during transcription. You can see the polyadenylation during transcription. So that C-terminal domain of our RNA polymerase is responsible for carrying out some of these processes. There's many repeats of a 7 amino acid sequence on the CTD that will bind to these enzymes that we need to be capping, splicing, and polyadenylating. These mature mRNAs now are able to leave the nucleus, and they're able to be translated. The export occurs through nuclear pore complexes, or NPCs, and these nuclear pore complexes only allow mature mRNAs to exit. Non-functional, not yet completely modified mRNA transcript can't get through it, which gives you another layer of control of what is going into your cytoplasm to be transcribed, to be translated. So some of this control we have is that there's a nuclear RNA export factor 1, or NXF1, that is going to bind to your cap binding protein, or your CBC. So here, your CBC, your cap, is going to bind to this nuclear RNA export factor, and that lets the RNA exit the nucleus. Without that 5' cap, the RNA wouldn't be able to bind and exit. And then once they're in the cytoplasm, most mRNA molecules are going to have a high turnover rate. That means your genes are constantly being transcribed. So the RNA that you have there is constantly being degraded, broken down, and replaced with new ones. We can measure them by half-life, which is how long it takes for half of the molecules to degrade. In a eukaryotic cell, the half-life could be several hours up to a few days. In bacteria, the half-lives are a lot smaller. They might just be a few minutes. mRNA can be synthesized again and again from one piece of template DNA. So you can amplify your genetic information, increase the transcription to have lots of copies of RNA being made. For an example, in your silkworm, it only has one copy of the fibroin gene, but you can make up to 104 copies of the mRNA from that gene. All right, so by now you can tell me about how RNA molecules, how the mRNA molecules are modified co-transcriptionally, how that impacts its ability to exit the nucleus, to interact with the ribosome, how that impacts having different versions of proteins being made from one gene. And let me know if you have any questions, and I'll see you next time. Bye.